My name is Michael Cook, a viticulturist with the Texas A&M AgriLife Extension Service. I'm also a full-time PhD student in the Department of Horticultural Sciences at Texas A&M University. It is an absolute privilege and honor to be a recipient of this year's American Wine Society Education Foundation Scholarship funded by Banfi Wines. I want to thank Banfi for their support, also for the scholarship committee rather, and all their hard work in having to go wade through all the applications of talented students. Also, I want to thank the members at large. Without your support, I would not be able to do what I do now, which is remaining full-time in my extension work, supporting our grape growers, not only in Texas, but serving on multiple national boards, supporting our American wine industry at large, but also being a full-time PhD student, traveling many hours every week for classes and for my research across the entire state of Texas. The research I'm working on, I'm very excited about, focusing on improving water use efficiency through irrigation strategies and cover cropping across 3 regions in Texas, while attempting to promote wine stability and red wine cultivars, phenolic profile, but all the while ensuring grapevine resiliency. Amidst climate change and the challenges we're facing in our American wine industry, we want to make sure that the practices we're utilizing in the vineyard promote a sustainable wine industry, production, quality, year after year after year, and that remain economically profitable. So I'm very excited. This is the first year of establishing the trials and running the treatments. So more updates to come, but I want to thank the American Wine Society Education Foundation and all you do, not only for me, but also for all the other students that have been recipients of these scholarships. They make a huge difference in our journey. And I want to thank you for supporting the next generation of viticulturists and enologists in the American wine industry.
This morning I'm at a vineyard in Hinesville, Arkansas. Right now I'm standing in front of Chambourcin vines, and I'm actually harvesting the Chambourcin for one of my research objectives, which is looking at 3 different strains of Lachantia thermotolerans, a non-Saccharomyces yeast. I'm evaluating how these 3 different yeast strains affect the acid chemistry of the Chambourcin wines during fermentation, and then I'll look at volatile compounds and how the colors change during storage for the wines. I am currently in the works with creating a grape production guide in conjunction with some other faculty at the university. We set out different standards for how to set up a vineyard, how to manage a vineyard, how to keep up the health of a vineyard, and this can be used, of course, by current grape growers in Arkansas and surrounding areas with climates similar to Arkansas, but we also hope that this production guide will be utilized by people who want to get into the grape industry. Another part of my research involves assessing color and phenolics of wines produced from co-fermentation of Noble and Merlot grapes. In the South, we grow mostly native and hybrid species because of their high tolerance to pests and disease pressures. However, wines from these grapes can produce phenolic-deficient wines with poor color stability over time. Muscadine in particular is an economically viable crop in the South, but it does have anthocyanin instability with brilliant color in the beginning, which can degrade over time. So the purpose of my research was to co-ferment Merlot and Noble grapes to enhance color, aroma, and flavors, and overall quality. My goal is that this research will aid growers and winemakers in producing muscadine products that are high in quality with great color potential over time. I want to give a big thank you to the American Wine Society Educational Foundation, funded by Virginia Wilder. This scholarship is really important to help support my research, and I look forward in the future collaborating more with folks in the grape and wine industry.
Hi there, my name is Nicholas Wilson, and I'm a PhD student at the University of British Columbia. Here in British Columbia, we have been experiencing more frequent and more intense heat waves, and this is extremely concerning for our grape and wine industry. For example, during a heat wave in 2021, several important wine regions here reached temperatures above 45 degrees Celsius. I'm currently standing in the UBC greenhouse where we are growing grapevines to use in experiments, hoping to solve the viticultural issue of high temperatures in BC. There are many different methods being suggested to growers for solving this high temperature issue, such as increasing irrigation, use of foliar sprays, and selection of different varieties. and these are exactly what my PhD research will focus on. I will be conducting experiments in controlled growth chambers to determine varieties typically grown in British Columbia that perform well under heat stress. I will also conduct experiments in British Columbian vineyards, where I will be applying heat waves artificially using miniature greenhouses and then testing different mitigation techniques such as foliar sprays and varying irrigation levels. I'm happy to say that we've been making very good progress in the short time that we've been working on this project. With this research project, we hope to ease the worries of grape growers when it comes to heat waves by finding practical and effective solutions to dealing with this type of stress. As I grew up in an agricultural town in British Columbia, I have an understanding of how stressful variable climate can be for growers. While we do not grow grapes where I'm from, I still find it extremely exciting to help ensure that the grape and wine industry is secure for the future. I would like to thank Castellarin Lab, where I'm currently conducting my research, for all of their help with this project, as well as their invaluable advice. I would also like to thank the American Wine Society Educational Foundation for giving me this platform to speak about my research. I strongly believe that with proper knowledge, the North American grape and wine industry can be prepared for a changing climate.
Hi, my name is David Campbell. I am a PhD student at the Penn State Department of Food Science, and I'm going to be presenting a research summary as an American Wine Society scholarship recipient. My research is focused on Pennsylvania wine grapes, so a few facts about our industry. It is a growing wine region. It recently rose to 4th in the nation in total wine production, and it provides a significant impact to the state's economy, something around $7 billion in total economic impact. However, Pennsylvania has some characteristics that are suboptimal for grape and wine production, namely very cold winters and warm, humid summers. This makes it important for grape growers in Pennsylvania to utilize hybrid grape cultivars. Hybrid grapes are robust to a variety of adverse weather conditions, pests, and diseases. However, they have important chemical differences between Vitis vinifera cultivars like Cabernet Sauvignon or Merlot, which may affect wine quality. So our research is focused on these chemical differences. An important one is hybrid wine tannins. So wine tannins are important for the sensory characteristics of a dry red wine, and they are known to be very low in hybrid grapes. So we are focused on tannin extraction dynamics, tannin structure, and how these correlate with grape cell wall components. Additionally, Pennsylvania is a very large and diverse state, both in geography and in climate. These are things that are known to impact grape and wine chemistry. However, we don't really know how they affect Pennsylvania's grape and wine chemistry. So a lot of our research is focused on examining how different location effects can impact our grape and wine chemistry. I would like to thank you for your time and attention, and I would like to thank the American Wine Society for selecting me as a scholarship recipient.
Hello, my name is Equine. I'm a PhD candidate at Penn State in the Department of Food Science. My advisor is Dr. Misha Kwasniewski, and I'm a part of a wine research lab here on campus. I would like to first thank the American Wine Society and the Education Foundation, especially the Pennsylvania Lehigh Valley chapter, for awarding me this academic scholarship. My research is aimed at developing an alternative method for aging red wine. Traditional aging methods, mainly barrel aging, is quite costly, time-consuming, and not very predictable. During wine aging, very small amounts of oxygen are incorporated through the pores of the barrels, oxidizing alcohol into the compound acetaldehyde. This compound then reacts with tannins and other compounds, including color pigments in the wine. This reaction will then bridge them together, causing elongation. Overall, this reduces the bitterness and sandpaper-like qualities of wine, while also increasing the color intensity. My work skips over the first set of reactions by adding food-grade acetaldehyde, which is in many foods we consume on a daily basis. Using acetaldehyde at levels that is in other foods can allow for the same characteristics that we see during wine aging at a fraction of the cost and time with more predictability. Here's an example of what adding acetaldehyde can do for a wine. These are model wine solutions containing tannins. The darker sample is where a colorless acetaldehyde has been added, showing that the reaction has been taken place. Following this, here are two samples that have wine color pigments extracted instead. Again, the darker one has the acetaldehyde added, which had reacted to create an increased color intensity. After characterizing these reactions, we then created an analytical method using this machine here called the LCMS to measure the products of these reactions. Not only does this help us monitor during the trials, but it can potentially be used in the industry to measure the progress of traditional aging methods like barrel aging. Thank you so much for listening. And again, thank you to AWS for this academic scholarship.
Hi, my name is Emma Gilbo. I'm a master's student at Penn State University, and I'm conducting 2 research projects with the goal of mitigating spring freeze injury in North American vineyards. Spring freezes are capable of damaging or even killing vulnerable tissues, which can later affect aspects of harvest such as juice chemistry and yield, and ultimately impact North American vineyards and the wines they produce. In the winter, grapevines become dormant to protect themselves against cold temperatures. Then, in the spring, they undergo a process called deacclimation, where they gradually exit dormancy. In my first study, I'm looking at the relationship between deacclimation and something called chill accumulation, or the amount of time that vines spend exposed to certain cold temperatures. I am evaluating how this relationship may change at different vineyards across Pennsylvania over consecutive years, using samples from 2 popular wine grape cultivars. Better understanding of these dynamics can help us more accurately assess spring freeze damage risk. For my second study, I'm exploring a potential strategy to help mitigate spring freeze damage by delaying bud break, or the time when vulnerable tissues become exposed in the spring. Delaying bud break can shorten the amount of time that fragile tissues are at risk of being damaged by spring freezes. I am testing whether a synthetic analog of a naturally produced plant hormone can delay bud break in a popular hybrid vine. I'm also evaluating other effects of applying the hormone analog, such as whether it impacts naturally produced levels of the hormone, yield, or juice chemistry, to get a better understanding of its effects when applied to grapevines. Overall, I hope that my research can help provide tools to North American growers to protect their vineyards against spring freeze damage and preserve the vines and the wines that we love. Thank you so much to the American Wine Society Educational Foundation and the Dayton and Springfield, Ohio chapters of AWS for your incredible generosity and support.
Hello everyone, my name is Walt Sanders and I'm a graduate student at the University of Georgia, currently working on my master's degree in plant pathology. Now right now, I'm at one of my favorite places to be, and that's at our experimental vineyard in Athens, Georgia. So here we conduct a variety of research on horticultural practices as well as disease management strategies so we can hopefully give better recommendations to our wine growers who mostly inhabit the northern Georgia area. My research focuses on powdery mildew, which is considered a problematic fungus anywhere where wine grapes are grown. It is extremely aggressive and can turn grapes from this into this in just a matter of weeks. Grapes infected with powdery mildew have very low sugar and water content and are overall unfit for any sort of vinification. That's why my research focuses on increasing the efficacy of some of our fungicides that we utilize to control this very problematic organism. More specifically, I'm looking into increasing the efficacy of elemental sulfur, which is one of the world's oldest fungicides, as well as one of the most frequently used fungicides in a vineyard setting. I'm in one of our chemical rooms right now, and you can see the sheer amount of this fungicide that we keep on hand. The reason being, we simply need that much to control for powdery mildew during the season. Now, my research is looking at incorporating an agricultural surfactant into each of those sprays. An agricultural surfactant is simply soap that you add to the spray tank, and it's supposed to give a more even wash of the sulfur across the leaf surface, increasing coverage and therefore potentially increasing efficacy. Powdery mildew is just one of many diseases growers face here in Georgia and around the world, but researchers tend to take problems one at a time. Thank you for listening, and thank you to the American Wine Society for supporting me and my education. Thanks.
Hi, I'm Sydney Rogers, a second-year viticulture and enology master's student at UC Davis, and I want to be talking to you a little bit about the work we're doing here in the Montpetit lab. Climate change is posing new challenges for the wine industry everywhere, including California. As temperatures rise, grapes are ripening faster, which could cause higher sugar levels, lower acidity, and more unreliable fermentations. But what if we could adapt to these changes naturally? This is where native California yeast strains would come into play.
Here at the University of California, Davis, in collaboration with Toomey Winery, we are studying a unique group of yeast strains found naturally in Toomey's California vineyards. These strains, called Pacific West Coast wine yeast, have evolved from native California oak yeasts. Unlike commercial European strains, these native yeasts are likely better adapted to the changing environment in California.
We will be conducting experiments to see how well these native strains can ferment and their impact in winemaking. First, at a smaller scale on the benchtop in the lab, and then at production scale using Toomey's Pinot Noir fruit and these specialized UC Davis research fermenters. Then, we will assess their impact on wine chemistry and wine flavor, aroma, and overall quality. Early findings suggest that the native strains can outcompete commercial strains in a fermentation while altering acid chemistry. These native yeast strains, seeing as they are already adapted to the California environment, could help provide more reliable fermentations while enhancing complexity of the wines produced. This is especially important to winemakers who want to preserve a site's terroir by using its own native yeasts, and to consumers like those of newer generations who prefer environmentally conscious wines.
By harnessing the potential of these native yeast strains, this research could contribute to the advancement of sustainable winemaking practices and the productions of wines with unique characteristics tailored to consumer preferences.